Patrick Bell, and this is the central chapter of the Oklahoma Native Plant Society. We try to get together once a month unless we don't. And we've got a meeting in November. We're going to take holidays off, and then we've got all kinds of exciting things coming up next year. After the January break in early February, there's a guy that's going to be coming into town by the name of Douglas Talman. If you're not familiar with him, he's kind of the evangelical bringing nature home guy that talks about why it's so important. We study native plants in the Native Plant Society, but his message is the importance of native plants as far as habitat and insects and biodiversity. And if you like watching your bluebirds, they got to have something to eat, and what they eat are the insects that grow on native plants. So anyway, Talmy is coming to town. Talmy is coming. I like that too.
Oh, through Christmas.
Okay. Okay. Well, you got to give a guy a break when you can. So anyway, Talmy's coming in February. As a prelude to that, we've had meetings every month. This is now the third in a row, talking about what to do with getting native plants going, either converting or changing your habitat. And tonight, we're very excited to have Jenna Messick, who is the Herbarium coordinator, caretaker. What is it that you do?
I'm the curator.
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 Apologize.
So I make sure we keep it all in order.
Okay, as well as soon to be associate professor at UCO. And she's going to talk about native plant seed germination, what works and why. It's important to understand after she concludes this, we're going to have an election for a central chapter chair. It's very important that you understand that if you leave, you'll be nominated. So everybody hang around. It won't take very long, and then we'll get this over with. Jessica, thank you for joining us.
Thank you. Okay, hopefully everybody can hear me okay. This is actually a bigger classroom than I'm used to at UCO. But yeah, so Patrick asked me to speak about native seed germination, but I just want to kind of give you a point of where I'm coming from before we really dive into some things. And of course, I always forget. So I'm from Oklahoma for most of my life. I did all my degrees down at OU. Okay. So I've spent a lot of time in the herbarium down there. I was in the Department of Microbiology and Botany when it was still called that. If you all remember that, because they've changed it now, it's plant biology. But then for my master's and my PhD, I hopped on over to the Department of Geography and Environmental Sustainability and came at it plant world from a biogeographic perspective. And then I stuck around OU a little bit longer because it's Norman. I love Norman. All right, did my postdoc with Dr. Moore, who's the current curator of the herbarium down there in Norman. And then had to leave the nest, right? So one of my criteria and moving on is I really wanted to stick with being involved with native plants and specifically with herbarium work. And so UCO needed somebody to do just that. And so that's why I'm there. And this is halfway through the semester of year 3 now. So it's pretty cool. Been around plants literally my whole entire life. As soon as I could walk, my grandma had me in the garden doing everything, pulling weeds, planting seeds, getting rid of various caterpillars and bugs because, you know, those were her prized tomato plants. But then as I got older, I started, you know, going out on my own and what kind of plants I wanted. And, you know, while I was still in school, I was like, plants are really cool, but what kind of cool, really cool, weird ones can I get? And so I started mostly just getting cuttings from people. And then I switched to germinating seeds because poor college student, it's way cheaper, right? But you got to play the long game with seeds. You got to have some patience first to get them to germinate and then let those plants grow. And one of the first really weird plants I decided to try to just germinate from seed was a Ipomea platensis. It's a little native morning glory to South America. It's a perennial. And so this is a picture of what their seeds look like. This is the plant. Notice it doesn't even have normal morning glory shaped seed or leaves. They're like all fingery like, but that's all what it looks like in highlight. You put this plant in lower light and then you see that normal heart shape. Okay. But one really cool thing, what really got my attention with this particular species is it does this awesome cudiciform base, and you can bonsai these. And this year mine didn't produce as many seeds as she normally does because I made a no-no and I planted too many other vining things and there was too much pollinator competition. So hopefully next year I'll have another round of thousands of seeds because it's really fun little plant. And then from this plant, things just took off for me, especially when my baby brother came back from a trip to Hawaii and brought me bird of paradise seeds. Those were challenging to get to germinate. I was like, what the heck? You know, they're super neat looking seeds. I love the little orange mohawks on the seeds. But he brought me a pack of 5. I have one. It's now in a big old pot. It's 14 years old. Right? So it's playing the long game. Hasn't flowered yet, but hopefully next year, fingers crossed, right? It's always fingers crossed. When are you gonna? Okay. Then this on that same trip, my brother brought me back a cutting of a plumeria because he knew I liked the plumeria. I had one until it froze in Tulsa in 2007 because I was up in Tulsa then. Apartment was below freezing for 9 days. I lost a lot of plants. So it was a really sad time. So I ordered some plumeria seeds. All right, and this is what the little seeds look like. The little wing is it's weird. You plant it, you leave the wing sticking above ground. You just have the seed barely below the surface. And that's Mary, that I named her. She is also now 14. She just came in last week because she doesn't like it when it gets down into the 40s. And again, when are you gonna flower? I've been waiting 14 years for this tree to flower. And next year she's gonna be too tall for the ceiling. So I'm gonna have to make some cuttings. But I'm hoping it's some really pretty colorful flower because the only way to get color diversity in the plumeria is to grow them from seed because all the ones with the Hawaiian lays are all usually taken from cuttings, so you get the same color of flower consistently. So maybe next year. Okay. And while I was on the website ordering plumeria seeds, I was like, why not try baobab? Why not, right? So I know these aren't natives, but at the time it was all about what's really, really cool. And so baobab seeds are actually pretty easy to germinate if you can get them. I grew them. I got a pack of 5, 2 germinated. So I thought that was pretty good. Then they got really big, like 3 feet tall. And I was like, I can't handle you anymore. So I gave them to a friend who's got them at her house in Arkansas. And last I heard, they're like 7, 8 feet tall. So they're still doing well. But that's, kind of where when the germination bug really caught me was all the weirdos. And I was like, well, you know what? I'm in Oklahoma. I really probably shouldn't be planting a whole lot of weird stuff that doesn't really do well here because I know Mary's getting big. I have a dolly just to bring her in and out every season. I even have a ficus, the ficus benjamini that was my grandmother's. He's at least 55 years old now, and he also requires the dolly. So it's getting to be backbreaking. So I'm like, all right, let's maybe start mingling in the natives because they can just stay outside, right? So I've gone on a lot of native seed germination adventures. And I really, really got started when I was finishing my undergrad at OU. And so right at the end, finishing my bachelor's degree, I got presented with the opportunity to do a master's over in geography, but not botany, with Penstemon oklahomensis, right? There's several populations at Tinker Air Force Base, and their biological management team wanted to make sure they didn't destroy those few populations that they have on base. And that was really cool. And I was like, great, they're going to pay for me to do research for a master's. So I was like, yeah, let's do it. So the summer in between undergrad and master's starting, that's when seeds are ready for Penstemon. So had to go out and do all this work and ended up doing 2 years of germination trials and found some really interesting results. Because Penstemon oklahomensis, if you aren't super familiar with it, was considered endemic, only found in Oklahoma up until 2010. Only in Oklahoma. That's its name, right? Oklahomensis. So in 2010, some nice folks in Texas found a population that got across the Red River. I have hypotheses of how this happened. I can't prove it yet. But there's some other, now there's actually a second population in Texas. So it's spreading. We'll see. But back to the germination though. Tiny, tiny little 1 millimeter by 1.5 millimeter seeds. I sat there and counted thousands of them. And the first summer, and there was like 8,000 some odd seeds. The second summer, there was like 15,000 seeds. But the first year I germinated 1,200 seeds, or tried to germinate 1,200. And I had a set of seeds I put outside where I wasn't going to tamper with them. I was just going to let nature take its course and see what happened. And then I had a set, I was like, okay, I'm just going to put you in your soil. I'm going to put you in the greenhouse and water you. Cool. Took another set of the same number, put them in the fridge for a month. Took the second set, or fourth set actually, put it back in the fridge for 2 months. So the ones outside, in October, that same year after collecting the seeds, 7 germinated. I'm like, are you going to make it through winter? And are any more going to germinate? And the ones outside, I even built a frame with a wire across the top, because I don't know if you're real familiar with the south end of Norman's campus, right along Highway 9. There's all those big greenhouses that they set up strings in. But outdoors, there's like big open areas and geese come in. So I was like, okay, I'm going to protect you at least with a wire frame. So I didn't need to protect them from the geese. I needed to protect all those seeds from the ants, because no more germinated. The ants stole them all. I was like, dang it. But the ones just in the greenhouse, I got 20-ish% germination. It wasn't super great. The ones that were in the fridge for 30 days, got 45% germination. All right, cool. All right. And then the ones in the fridge for 2 months, like 85% germination. Right. So I just put, and I even had trials of just regular, garden variety potting mix. And then I even collected some native soil from an area a little bit away from the populations in Norman, and got really close results. So they don't care about the soil. They just would care about that cold to get the seeds to germinate to really high numbers. And even after they germinated, they grew like their normal 2 feet tall. They flowered in the greenhouse for me, and it was great. Then I had to do it a second year, the same thing. But then I was like, well, okay, I got a whole bunch of seeds left from the first year. Let's see what a year in the desk drawer does to them. And I got flipped results. The 30 days germination, or 30 days in the fridge, actually had better germination, and then 60 days had way fewer, like maybe 20% germination. So letting them sit for a year was a good thing for some of them, but not too long in the fridge. Like a month is good. So we surmised and came up with a plan for Tinker. They're like, hey, if you want to keep perpetuating populations and collect seeds, but want higher than outside germination rates, if you collect the seeds that year, put them in the fridge for a couple months. But if you hold on to some extra seeds next year, you only got to wait 30 days. So if you want to just split the difference, say 45 days and be good both years, and all the years into the future. And so that was really fun. It was a big project. Besides just germinating the seeds for the Air Force, they wanted to know about habitat. They wanted to know about potential other locations. So I did a bunch of other research with this plant. Still near and dear to my heart. I finally got some new fresh seeds that are going in the garden this fall, because I miss them having them in the yard. Okay. Other plants I germinated right after I did my master's. I was working on my PhD. We were like, my advisor and I were just like, well, there's some Lauraceae family members in Costa Rica. He's like, but we have a Lauraceae member, sassafras, here in Oklahoma. Let's do some germination with that and see what happens. With sassafras, Forest Service has done tons of germination studies. 35% germination is the best they've ever done from the center of the range. And so sassafras grows, the furthest west it comes is Lake Holdenville, Oklahoma. That's it. It doesn't go any further west. It goes all the way to the East Coast, goes up to Canada, down into Florida, no problems. And so all their trials were from the middle of the range. Really, well, plants act weird on the edges of the range, so let's see what's happening with the ones in Oklahoma. That was fun. They smell so delightful when you're taking that little purpley skin off the fruit to get the one little seed out of there. And they look like, if you cut a fresh one in half, it looks like an avocado, but in miniature. But they're related, right? They're in the same family. And so I didn't want to like tamper with like, a lot of temperatures. I was like, I just want to know what's naturally going on with natural stuff. So we set up a big germination trial in a cold frame, again, at that aquatic or that area down on the south end of Norman's campus. But instead of a wire frame, I did an actual cold frame. I was like, all right, filled the seed trays with vermiculite, because I was trying to be kind of sterile, worrying about fungus, and put them in there. I was like, all right, I'll just come water you every other day-ish, unless it's snowed in or iced in. Otherwise, I'll come water. Right. Only 13% germination from the western edge of the range. Did it a second year, just to be sure, 13% germination. So sassafras trees love to send out suckers from the roots. They'd rather reproduce vegetatively in Oklahoma, is what we surmised. And so there's one of the little baby ones. I gave them all away. The first year ones, the second year ones, there was an incident with some non-native snails that got introduced to my neighborhood. So they came and ate all the baby sassafras trees. They ate my aloe. They ate the paint off the shed. Yeah. So Dr. Berge down at the biosurvey in Norman, her and her undergrads came and did a whole study in my neighborhood. And they found the epicenter. It was a neighbor. But they came and ate a lot of my plants. So maybe someday I'll try germinating sassafras again, just because they're fun. I like the way they smell. They're really nice little trees. At the end of the sassafras germination, though, my advisor, he's like, hey, I collected some pawpaw seeds. Stick them in that cold frame just because you got room. I'm like, okay, right? He gave me 14 seeds. All of them ended up germinating just by being in the cold frame, getting watered every so often. Just being out with, the winter temperatures. Didn't have to do anything special, which was really nice because they have a pretty thick seed coat on the pawpaws. Then later on, get into my postdoc because, school takes forever. Was looking at some genetic stuff with Salvia azurea, grandiflora, or variety grandiflora. Because why not? It's got a really huge range. There's 2 varieties across its range. There's kind of this cool biogeographic barrier between the east and the west called the Mississippi River. It was like, hey, maybe something's going on there. But as a side project, just for fun, because I was so into just looking at this plant over and over and over, I'm like, I found some seeds. I'm going to see what happens. And I threw some in my fridge. And after 6 months, some of them started to germinate. I was like, 6 months is a long time. Maybe I left them in too long. And so when I got up here to UCO, I had a student interested in this plant. And she wanted to try to germinate it herself. I was like, yeah, sure. Here's the supplies. Here's some locations that you can collect some seeds, but don't go crazy because we need them out there in the world. All right. And so we ended up doing 3 treatments. One where we just put them in little dishes with vermiculite, kept them moist, throw them up under some lights on a timer, put some in the fridge for 30 days, put some in the fridge for 60 days. And that 30 and 60 days is a pretty common number you'll hear, right? It's pretty common because it's easy to remember 1 or 2 months rather than 45 days. And so turns out with the Salvia azurea, it really, really wants the 60 days. We got like 80% germination, 75, 80%. It was really high. The ones that had only been in the fridge for 30 days, hardly any. And then, so after waiting, the student got really impatient and put them back in the fridge for another month, and then they all germinated. So a lot of our native plants need cold is a common theme you'll hear as well. Okay. So other ones that I've been working with students on up there at UCO are, so here, this is the Phacelia integrifolia, super common on the gypsum outcrops at our field station. It's not found off of gypsum soils very often, right? Its closest genetic relative, the Phacelia conjusta, is found on limestone soils in only 2 counties in Oklahoma. So we did some comparisons for germination and growth with and without gypsum added to the soil. And then because we're at the field station, there's another gypsophilic plant, the little Nama Stevensii. We got seeds of that, and we tried to germinate those. The Phacelias don't really care. They're ready to germinate almost immediately. We forgot to put some in the fridge right away, and they're like, a week later, okay, cool, we're happy to germinate. But putting them in the fridge for at least 30 days got higher percentages for germination. The little Nama Stevensii was the oddball out. We were just put some on a petri dish with a little filter paper, and we just put it under some lights. We're like, well, let's just see what happens, and then we'll go from there. And after about a week, like all 10 seeds germinated, and we're like, oh, well, this is going to be easy then, right? So we set up trays, put them in the fridge, and after 60 days, 7 out of 300 germinated. So we're going to go back to the drawing board. That student graduated, so I got to find a new one that's interested in trying this out with me. Otherwise, I'll just do it myself. I mean, I got a fridge, I got a lab, I got the supplies. I can't help it. I love germinating seeds. I like the challenge. I like to figure out what they need, especially if it's a species that doesn't have information all over the internet, right? Those are the ones I really like. So with those Phacelias, like I said, they really didn't care. So a lot of them are ready to go. They did really good after about a month. The 60-day ones didn't even make it to 60 days in the fridge. At 44 days, they're like, hey, we're coming out. You got to, so they're hitting the top of the plastic. We got to pull them out of the fridge, stick them out in the greenhouse. So a month really helps it increase the numbers. Yes.
Are these germinating in cold conditions? Is what you're saying?
Some of them will, yeah.
So
Yeah.
you're not talking about when you pull them out of the refrigerator and put them in the greenhouse. You're talking about in the refrigerated environment. They're
Some of them will pop. Some of them will go ahead and germinate. And once they do that, that's the sign it's like you got to get them out, because those ones that do come early won't do well after, because there's no light in the fridge. I don't think it, and I've figured out a way to light the inside of the fridge, but that would be a fun trick. Yes.
So what are they in the refrigerator?
I use just normal 72 cell flats. And sometimes I did splurge, and Johnny's seed catalog had some ones that had just lines rather than separate cells. And so we did some of those too. And for all of these, we didn't even mess with vermiculite. We went with a potting soil mix, but then we added gypsum to half of them. And we also added sand, because those gypsum soils are really, really sandy. And like we even sent a soil sample to Stillwater, and the calcium content from that gypsum was like almost off the chart. It was like 24,000 parts per million. It was insane. And that's like, you know, a really harsh environment for any plant. And these two on the far sides, they thrive in it, right? And I wanted to do a similar study on at the same time with this Nama, with its closest genetic relative that doesn't live on gypsum, but we couldn't find any seeds. I found them this year, so it's on. Challenge accepted, right? Okay, so those are the things I've been doing. I've got some other things I failed at completely and miserably, but that's okay, because I'm going to keep trying, right? I love Indian paintbrush. I really do. And I know it needs a host to go with it, right? And so I've tried numerous ways to germinate its seeds, and I haven't quite figured out what it needs. And I'm thinking it might be a fungal association in the soil. That's my next thing to try, because I've tried just putting it out for the winter, nothing happened. I've put it in flat along with other seeds that I thought needed the same cold requirement. So, hey, you'll have a friend. No. So next thing to try is, when I collect seeds, maybe take a little bit of a soil sample and see if there's some sort of fungal association that might help those seeds germinate, because some seeds do need that, right? We're gonna come to the far side, and then we'll come to the middle, because that one's a funny story, right? So down here, so every year, I'm a Cherokee tribal citizen, and so every year I get 2 species of seeds I get to pick on the website, right? And they mail them to me. One year they mailed me some jewelweed, and I was like, that'll be awesome, because we don't have that this far west in Oklahoma. And I'm just now becoming allergic to poison ivy. Like, okay, cool. You know, help with the itch a little. So they sent the seeds, put them in the fridge. Nothing has happened. So I'm going to leave that pot outside. It's covered to keep protected from the squirrels, because I have a squirrel problem. And so maybe after a second winter, maybe next year, I'll have some jewelweed. But right now, I'm going to say it's a failure, but we'll see. I'll try. I'm just gonna give it a chance. I mean, they might do it. Above it is Stillingia sylvatica, and this one I was asked to figure out how to germinate it by Cherokee Nation. They want it in the cultural gardens. This is a culturally important plant to the tribe. This one's tough. I have put it in the fridge for over a month. Took them out, warmed them up, nothing. I was like, all right, come on. It does have a pretty thick seed coat, so I nicked it, soaked it. Let them sit all this summer, watered them. They're in my craft room, which doubles as a plant room, because you know how that goes. You gotta have space for the plants too. Nothing. So they're back in the fridge. So maybe they need 2 winters, because I can't find anything really.
What's it called?
Stillingia sylvatica. Queen's Delight is its common name. It's in the Euphorbiaceae, so it's got that latex. So I gotta wear gloves. But it's a really neat plant, and I found this massive population this summer, and I was like, oh. So I was able to send a student to collect some seeds for me, because I wasn't allowed to do field work in September. I got my knee fixed, so it's the spring. I'm so ready. So failure for the first year on the Stillingia, and so one thing I want to try with, because I got some fresh seeds now, I'm gonna put them in the freezer. I'm gonna skip the fridge. I want to go straight to the freezer for a couple weeks, but I don't think I should push it past that, and then maybe back into the fridge, because maybe they need a really, really cold, and then a lot cold, but a little bit warmer, and then I'm gonna throw them on the heat mat, because these bloom late, or they start blooming June, and they're pretty persistent up until about now. But most of them, their seeds are popping off already, right, or gone by now. So it's a failure for now, but I'm going to keep trying because it's really neat. And like I said, I love the challenge of just germinating seeds. All right. So in the middle, that's our Salvia lyrata. That's only from the eastern part of the state. I'm from eastern Oklahoma. I like some of the eastern plants, and I want them in my backyard. I try to contain them. So visiting family out in Sequoia County brought back a couple of these because I teach plant taxonomy in the spring at UCO, so plant identification. And I brought some of them back for students to learn how to identify, because there's not any salvias up in the spring on this part of the state. And so I brought some back, and I was like, all right, let's identify them. And then, as with everything after the end of class, it comes home and goes in my yard. Everything I take to class for students to learn to identify ends up in my backyard. Even the dirt left in the trays, if they like completely shred the plants, because there's probably seeds in there, to really diversify my nice Edmond HOA backyard. And I put a lot of stuff in that yard because I want to irritate that HOA. You should see my front yard. I mow it just to keep them quiet and off my back. But so I put one Salvia lyrata in the backyard in the designated pollinator area, garden area, and the bunnies came and ate it before anything could happen. And I was like, well, darn it. So all summer, there's one persistent one of these in a completely different area of the yard. And I'm like, all right, so maybe Salvia lyrata needs bunnies to ingest them.
Oh, maybe.
Yeah, right? Because I've taken seeds from eastern Oklahoma and tried to cold stratify them in the fridge, and nothing.
Are you going to test that in the lab?
I would like to, if you can help me catch the bunnies. They're really cute little Easter cottontails. We have a bunny problem in my neighborhood, and I'm okay with that. Except for when they eat my plants. So a lot of things are in cages, or as I call them, plant jail. So I'm now getting Salvia lyrata spread around in this one spot in my yard. I'm like, cool. Now if they can get the birds to stop bringing the pokeweed, we'd be really good, because that's taken over my designated area. So this one's both a failure and a success, accidentally. I do like the bunnies. They're fun. They do things for the yard. So coming up though, I've got some ideas, because Alaska came to visit us in February. It was too cold, right? Killed all the non-native stuff, which was great, because except for the yard work afterwards. Taking down 60-foot crepe myrtles was super fun. And that poor Fatina, we need a bigger chainsaw at my house. So I want to take that Phacelia conjusta that was, hey, I'm ready to germinate no matter what. And I want to stick some of the seeds in the freezer for a week, because we got to almost -20. And that's about what I can get one of my freezers in my lab to. Because when I went out to collect fresh leaf material for my genetic project that I also have going on with this particular species, this plant, I couldn't find it. From all the historic locations in Comanche and Caddo counties, I can't find it. So I want to know, what did that cold do? So that's why I'm going to retry this one, but a totally different set of parameters. And again, I'm keeping working on that Sphalangia because I've been asked to. Okay, so those are some future things. But also future things, I like Indian blanket as well. I really want it in the yard, right? And again, irritate the HOA. I want some buffalo gourd. I've got one spot on the side of the house that I think it would be perfect. Plus, I don't really want to mow in the backyard. So, okay, right, natives. I've gotten a bunch of the Monarda fistulosa, the bergamot seeds. I like it just because it smells good. So I want to try it. I might have a student help out with it, or I might just put it in the garden. It depends on if I find somebody interested in helping me and that, gets excited about seeds. And also, I've collected seeds of regular old clover. I know it's not native, but it's the sacrifice to the bunnies. It is the sacrifice to the bunnies, because I found if I keep them busy, with something extra tasty, they will leave the other things alone. So I got to figure out some seeds germination for the backyard real soon, because we're coming into fall. As I plan the shift and backbreaking work to bringing outside plants in, I'm also thinking for the spring and the summer. Okay, so when you want to go and start planting from seed, because that's the long game, it's not as easy as just going and buying plants that somebody else germinated. You got to think about what species you want. So it's going to be the right seed. Okay, and then also you got to get the seeds out at the right time. So you got to figure out what they need. Okay. Also, you got to give them the right spot, because if it's not a spot when they're growing and mature that makes them happy, you're not going to be, even if you're successful on germinating them, if they're not living and thriving and ready to make seed again for you the next year, what's the point? Right? You can think about things like that. So when you think germination, technically germination is just when the little roots, the little radicles emerges from the seed. And if you do them in petri dishes, in the fridge, at home, or in the lab, it looks like this, right? That's considered germination. You don't even see any of the little cotyledons, the green leaves, right? Or here's some of the Facelias we played with. They're ready to go in the lab. But when we come at it from the gardening perspective, we're like, we want this, right? But then they're so tiny, you're gonna like, wait, where are they? Right? And then you gotta zoom in, and then your camera's all pixelated, and you're like, but it germinated, right? But technically, it probably germinated several days before it was to that point. Okay. So one thing also about the right seed is where your seeds come from. So several years ago, the U.S. Forest Service proposed these provisional seed zones for the lower 48, and they divvied up the continent, our part of the continent anyway, into what they call provisional seed zones. And there's a whole mess of them, as you can see, right? But they're based on climate and ecoregions. So then the idea is, if the seeds are sourced from an area, from a climatically similar area to where you want to put them, you're going to be more successful, right? So if we zoom in on Oklahoma, we only have 11. So it gets a much smaller area to think about, and then we throw in our counties, we're right smack in the middle. So a lot of this, I think this works. I don't use it very much, but yeah, this right here, all this is actually similar to all this stuff out here in Texas. So all seeds sourced from that same area theoretically should be acclimated and ready to do well if you keep them within that same zone. And we've got divisions, for eastern Oklahoma, because we got this precipitation line, right? Eastern Oklahoma, it rains a lot more, except in these last few anomalous years, like this summer. That was weird, all that rain out in the panhandle. But so that's the idea. So if you're thinking about seeds from that species might be native to Oklahoma, but if you're going to get seed from far, far away, it might not be as successful. It could be, but that's the idea behind the right seed, is trying to think about where your seeds come from and what their parent plant's climate was like. So seeds around here, why not get them around here and then put them out in the garden, make it way better than those ugly monkey grass plants, the stupid silver maples, the annoying plants that you don't really want to have to deal with anyways. So that's what I mean by when I say right seed. But also it's all about the right time, because no matter what, a seed has some level of dormancy, whether it's just a few days or years, decades, centuries. The time varies, right? Because if the seeds weren't at least dormant for a little bit, they would germinate right there inside the fruit. So there's always some level. And the reason the seeds can be dormant is because of something internally, right? Maybe the embryo is immature, just needs some more time to develop inside before it's ready to germinate. Or it could have something on the external aspect of it. So those thicker seed coats or extra things on the seed coat that get in the way and slow down germination. And on those external things, it could be even more than just the physical aspect of the seed coat. It could be chemicals that are hanging out and preventing the seed from germinating. So you got to break that, right? And sometimes those seeds need some after ripening, which kind of goes with the internal dormancy, where you just got to give it a little more time to continue to develop, because once the seed is left the parent plant, it doesn't stop doing its thing, right? That little embryo is still developing and then waiting for the right time and the right conditions, right? And so having to break that dormancy is super common in all of the temperate areas, which we are smack in, right? We're almost in the middle of a large temperate area. And so really, it's just all about preventing that little seed from germinating too early, little young plantlet, as I like to call them, you know, very tender stage of life for any plant, and you don't want them to die in the winter. Some of them can make it through winter, but not all of them. Okay. So, and you don't want them to come out, you know, and germinate too terribly early in the spring in case we get those weird, crazy late freezes. So here, sassafras seed. Right? You know, my whole big project, we had to, the ones that didn't germinate, I cut them in half to see if there was even an embryo to see if they were viable, and maybe they just needed more time. And unfortunately, viability testing kills them. But the stain, it takes that pink, and that embryo is very, very immature. So maybe just needed a lot more time for those seeds to, the embryos to develop inside those seeds more. So then maybe I could have gotten higher germination, but again, my goal was just let's see what happens, right? All right. So also that right place, you know, you can throw native seeds mostly out in the yard as long as it's going to be the right environment once it's a mature plant. And that's the easy thing to do. I don't know about you, but I like to have my hands on the roots. I like to plant them. I like to think about the space of how tall it's going to be. What are the color combinations going to look like? How do I prevent the bunnies from eating them? Plus, I figure if they're a little bit bigger, they got a better chance against those bunnies because they're ravenous. So you're going to think about your soil. I live in West Edmond. I only been up here 2 years. This is going on year 3. Should have done the soil test day 1. I just tried to do a vegetable garden the first year, and yeah, there was almost 0 nitrogen. I was like, dang it. And then I'm fighting the hose as well. The pH is 8.4 out of the tap. And I'm like, oh. So I have my work cut out for me. So I've been composting excessively since I've been up here from Norman. But you know, a lot of native seeds, they're not super crazy, or native plants are not super crazy. They don't need super high rich soil, but if it's already depleted and nothing is growing there, there's probably a reason. And so got to think about that when you're ready to start germinating seeds. Okay, so some basic things to think about when you're trying to figure out what to do to get your seed to go, right? So if it's got an internal something keeping that seed from germinating, and if it's physiological, it just usually the plant, the species need a period of warm and or cold with some moisture. And how long that period is and in what order, it kind of varies. And if you think about your spring flowering natives, they probably just need the cold. And then that warming up period that naturally would occur in spring. You know, I've read do this, this, and this to germinate this, this, and this seed. And the common theme is anywhere from 30 to 60 days. Is usually a good general rule of thumb. Start with 30 days, take some out. If they're not germinating, put them back in for a few more weeks. That's usually what works really, really well, especially on small seeded things that don't have a thick seed coat. Thick seed coat changes the game, right? And then you got to think about, okay, cool. And it doesn't hurt if it's a big enough seed to cut it in half and take a look at what that embryo looks like. How developed is it? Can you start seeing those first cotyledons? Or are they looking like that little pink blob on the sassafras? If they're looking like their little pink blob on that sassafras, seed just needs a little more time to let that embryo get more mature inside the seed. And then sometimes it's a combination. You got to have the time, you got to have the cold and or warm. Okay. If you're thinking about a plant that will flower later in the summer, it's going to need the cold to get through winter. You just think about that. But then it needs that extended warming up period. Because if they're not coming up even till June, July, and then flowering late in the summer, that kind of gives you clues just by looking at their natural flowering time. So then, you know, those externals, that's where we start thinking about those thicker seed coats. Scarifying them is a really easy thing to do. You take a pocket knife and nick and soak your seeds, or rub it on some sandpaper. I've done that with, I know magnolias aren't really native up in this part, but like magnolia trees. I like the way the flowers smell. Got some seeds, soaked off the red, pulpy, oily business that it gets the birds to come. And then took the seeds and rubbed them on some sandpaper, soaked them overnight, put them in a little tray of vermiculite in my fridge at home, because it was before I had a lab fridge. And 6 months later, 12 magnolia trees. And then Alaska came to visit. Now they're gone. So I'll try again. That's another failure, but it's, but it's an example of a seed that needs a little bit of scarification. And essentially all you got to do is get a little bit of a weak point so water can get in there and get the whole process started. And that's the goal with that. But some seeds have really thick seed coats will have those chemicals, and they're really, we call them chemicals, but really they're just hormones preventing that germination from happening. And what those seeds need is soaking and time, so that the water can naturally just pull out those hormones and help dilute them, so that then at the same time help soften that seed coat, and then germination can occur. An example of one it's another one I'm about to try on. I didn't have a chance to put it in the slides. Kentucky coffee tree. Somebody brought me some pods. I was like, okay, sure, why not? My list of to germinate, it gets longer every year, right? But I was looking on those. I'm like, those are really hard seeds. Nobody disperses them anymore because their dispersers have been extinct. And so I'm like, all right, well, maybe I'll do a hot water scarification. Pour some boiling water on them, and I looked it up. I'm like, cool, that's right. Sometimes I don't look it up, and I'm like, oh, I'll just try this. But sometimes I'm like, well, maybe I should check it, right? So that's what I'm going to do here pretty soon. And I have a lab fridge, so I won't get in trouble at home, taking up too much fridge space. But yes.
Did I put that right? You poured boiling hot water,
I'm going to. Yeah.
and you read that was the way to get them.
Yeah, Forest Service.
What in nature is going to boil those seeds?
Well, the Kentucky coffee tree probably had some megalofauna from the Pleistocene, so like 12,000 years ago, like large mastodons or mammoths may have eaten them and pooped them out. So that I'm kind of mimicking a speeded up acid process to help try to get the chemicals that delay germination on them.
Okay,
Kind of out.
so you do see them in the wild. I
Yeah,
mean, I've seen them a lot.
just give them enough time.
Okay.
Yeah, so
you
I
just
just
speed
try
it
to, yeah, I'm just gonna speed it up.
Okay. Thank you.
Yeah, because give it enough time to seed out of the environment. If it doesn't get eaten by something, a seed predator or fungus, give it enough time, it probably will germinate because of the constant precipitation coming down, raining, will help you wash away any potential chemicals preventing germination, but also weakening that seed coat so that this little plantlet can get out, right? They all want to get out eventually. So, but yeah, so there's tricks to like speed it up because I mean, yeah, I play the long game, but I'm not that patient. I mean, granted, my plumeria and my bird of paradise are 14 and no flowers, but that's different. With waiting for flowers, I can deal with because I like the growth watching. But seeds, I get a little impatient sometimes. And other external things to break. Sometimes you just need a combination of nicking that coat, letting water get in, and or getting the chemicals out of the seed coat, and I'll add in a period of warm and or cold. So it's not always just one thing. But sometimes it's a combination. And combination moves, well, they require a little patience, but they're possible. So when you're doing warm stratification, what I mean exactly by that is you have your seeds in some sort of potting medium. I really like to do vermiculite on seeds just because it's a little more sterile. There's less risk of fungal attack. I've had a lot of seeds just rot because that can happen. And also, it's a lot cleaner in the fridge than having just dirt in the fridge. And then, so warming them though, just it's like your regular vegetable garden. You put your seed in the soil, it's warm outside. A couple weeks later of watering, your plants should come up. The seeds will germinate. And it's usually, it's generally 60 degrees Fahrenheit. Sometimes some plants are cool with 55 degrees. And the warming period can be a few days to a month or two. And just give it time. Okay. But when you're doing cold stratification, you can do the same process. Put your seeds in a tray, whatever you like, whatever you got room for. Again, I like vermiculite. Sometimes I'll mix in a little sand You don't want to do, you don't always want to do straight sand because it doesn't hold water long enough. So then the seeds could dry out if you're not constantly making sure that moisture level is good. And then you stick them in the fridge on the lower part of the fridge, not the upper part. That's a trick, right? Upper part, a lot of fridges have freezers above there, and that's way colder. And then you could risk freezing and possibly killing the seeds if they get too cold. So bottom of the fridge, a little bit warmer, but still cold enough to mimic winter. And so usually, you know, if you, yes, I have a fridge thermometer, but you know, our fridges, we keep them, you know, roughly just below, above freezing. Some of them go up to 42. And I've been fine having a fridge thermometer, keeping the fridge for those patent specimens. We kept it like, I kept it like 40 degrees Fahrenheit. So, and that works great. So you don't want it too cold though. And no matter what though, with these methods, you've got to remember the moisture, because if they dry out, then you're kind of resetting. And then you gotta wait a little bit longer. There are very few species in the world. I couldn't find any reference to any that are native to Oklahoma that just cold, dry stratification would actually work. Because I've tried it on some seeds, and I'm gonna be like, okay, I'll deal with you later. I'm just gonna stick the packet in the fridge, and then I forgot about them. But that just puts them more dormant because they're dry. But there are a few species out in the world that you can do that. Just dry, cold stratify them, and then plant them, and they'll be fine. But none of that are native around here. So that's why I have not all species. Yes.
You say either way, don't forget the moisture.
Yeah.
What are you talking? Spritz them every day or?
As needed. So if I'm doing it in my lab because I can fit big trays in the fridge, we just put a saran wrap on the top to keep some moisture in after we get them set, and we check them like every other day, sometimes every third day, if they're staying moist enough. But if I'm doing it at home where I have limited refrigerator space, I'll cut the bottom of a milk carton, about 2 inches tall, punch some holes in the bottom. You set it up with the vermiculite, get the seeds in there, and I'll put it in a Ziploc bag and partially seal it to help keep moisture in longer, so that I don't have to check it every day, because I got lots of things I can do every day. And so, but yeah, and then I just spritz it with a spray bottle as needed to keep it moist but not soggy. Yes.
Yeah, as soon as my seeds they germinated, and the issue is not about germination, it's more for the squirrels. They keep digging the pot, and then so they were just kicking and until everything out of the pot. So I tried everything, you know, I tried to put heavy mulch, it didn't work, and then so I put like rocks on it, they would kick off the rock, and then I even used the expensive spray, and they will get on the part that the area that there's no spray.
Right.
So they are just raising hell everywhere they are. So,
I hear you.
what do you do to protect them?
So I found on the side of the road one time, somebody was throwing away their tomato cages, but they were ones they had made themselves. And so they were 1 inch by 1 inch wire, just rounds. So I put those around some pots that will fit in there. And then I throw wire on top. That's the same, roughly the same, about 1 inch by 1 inch, because that's the only size that the squirrels I've found can't get through. 2 by 2, they go right through. And so I literally put all my plants in jail for a while to try to prevent the squirrels. And then as the plants get bigger, I have a whole mess of garden rocks, because it's fun to go to Arkansas and dig quartz crystals. And I'll line the pots in rocks. But if they start messing with the rocks, I'll just sprinkle blood meal on the surface of the pot. So I'm adding nitrogen, but the squirrels don't like the smell. And it also keeps the stray cats out.
I'm sorry, what did you sprinkle?
Blood meal. Blood meal, just straight blood meal that you get at the hardware
Are you
store.
able to do just the blood meal without the cage?
Yeah, I do that because the squirrels that were trying to get into the bird of paradise pot, and I was like, and they were kicking all the rocks off because I did rocks first. So I started sprinkling blood meal, and they leave it alone. They don't. There's something about the smell they don't like. Yeah, and it got my wax foya, or my narrow leaf wax foya to flower doing that. Because I was like, why are you not flowering? And then I was like, I'm gonna give you some blood meal because I don't want the squirrels to eat you, because squirrels and I fight about my succulent type plants. But yeah, blood meal.
And second question, as far as monitoring the moisture
Uh-huh.
in the fridge, how do you monitor that?
Put my finger on it and see how wet it feels.
If it's moist, you just,
Then I just leave it for another day or two.
okay.
Yeah.
And do you put the seeds on the vermiculite or in the vermiculite, or?
Depends on how big the seed is. So if it's a large seed, I'll put it about twice the widest part of the seed deep. If it's a tiny little speckless, dust speck seed, just on the top. And that's why I cut when I do it in like the milk carton bottoms, I make it about 2 inches. So I only have about 1 inch, maybe 1.25 inches of vermiculite. So I have room so I can have some sort of plastic to help retain moisture without touching and possibly static clinging off seeds, because that sucks to lose them like that. I've done that on accident. Yeah.
The milk jug thing, I did this last year, the winter sown
Mhm.
method, and you leave the milk jug top on there. I mean, you cut it off, you cut it in half to plant your seeds, but then you duct tape the top part back on and leave the plastic lid off. And man, I got so much stuff to sprout, the native bird of paradise, all kinds of things.
Yeah.
With that winter sown method, there's a website, wintersown.org, that has great instructions, and that would probably help the squirrels too.
Yeah, to get them started, give them a
And you
little.
just make little mini greenhouses with plastic jugs, and you put them out. I watched a webinar from Texas, and they were saying they always do it on December 21st. But I waited because we had this horrible weather. I waited until that was just the week after that was over, that last Arctic freeze, and I had great results. Even old seeds that I've had a long time, I just put everything out there, and so I think that works.
Yeah,
You said that was winter stone?
Winter sown, SOWN, and not everything's outside. And so it goes through the freeze and thaw cycle of whatever's happening. And you do water it and watch when you see condensation inside the jug, you know it's got enough water. But when you don't,
give a little spritz through the top.
Mhm.
I've done that with some native seed mixes and had pretty good results. And then those snails.
Well,
It's a constant battle, right? If you want the plants, but there's things that want to eat them.
Do you know blood meal deters armadillos and or raccoons?
I don't know for sure. I've never tried it because there are raccoon feet back there now.
My damage is mostly.
But my plants aren't messed with, so maybe it's working. Because yeah, we did have raccoons show up over the summer, and last fall was skunks, so that was fun.
I was thinking they would like blood meal if it was one of those, because they're scavenger type critters.
Yeah, I haven't. But since I've been doing it, yeah. But yeah, since I've been doing the blood meal, I haven't had anybody messing with them, and I know for a fact because I've put the trail camera in the backyard. That's how I know those were skunks. And I see the footprints from the raccoons, and I need a new dog. I've been dogless for a while, and so maybe later this year I guess adopt a new one that will help keep things out of the yard.
Use them today, only where you want them to.
I had a Brittany that I did that. Yes, I was like, because she wanted to dig. I'm like, right here, and she's like, I'm like, yes. It was a win. It was a great thing. That was a good dog. Okay. So when you're to pick up where we were talking with the dormancy breaking, when you need to manually scarify those seeds, if you need to nick them or rub them on the sandpaper, you want to pay attention to where you're doing it. So if you this is just a typical bean seed for illustration, but there's this little spot right here where the radicel is going to come out, that first root. So if you're going to nick and then soak them, or if you're going to just use sandpaper, you want to stay away from that spot, because if you damage it, then you're going to probably not have germination actually occur. So where you do that manual nick or sandpaper rub actually matters. So, because the idea is to do it somewhere else, over in here somewhere, just so you can let water get into that seed coat faster. And so I call it the nick and soak method because that's what my grandma called it. I mean, you always have a pocket knife ready to do that. If you do blue bonnets like the lupines and those really hard, but they're small seeds, don't even try to nick those. They are stone hard. Don't hurt yourself. Just rub them on the sandpaper. Okay, so safety first. And the plants are pretty and all, but don't need to cut yourself. And I'm prone to cutting myself, so yeah. So you don't want to damage where that first root's going to come out, that little radicel, because we want the plants. Then dealing with those chemicals, like I was saying earlier, you can do a cold moist stratification and just given enough time, especially the thinner seeds, you're going to be able to leach out those chemicals and hormones, and so the seed will germinate. Just give it enough time. But if you want it to go faster, then you can bring the water to a boil, and then pour it over the seeds and let it sit overnight, even up to 24 hours, depending on if it's a really large seed, and that will increase your germination rate because you're speeding up that leaching process with the hot water. And then sometimes you got to do both. Sometimes you got to do both. There's not a whole lot of native Oklahoma plants that need both. Those Kentucky coffee tree maybe, if just, because I want to speed it up. And then it's all about the right depth, like we were talking about just a moment ago. You don't want to put your seeds too shallow, even if you're doing them in a tray to begin with, because you want them to develop a nice healthy root system. So if you plant your seeds too shallow, they're not going to be, they're going to be a weaker root system. They're going to be more surface, and you have less likelihood of like damp off, wilt, flopping over, not being a good healthy strong plant. If you plant it too deep, that seed is going to use, or little plant's going to use a lot of energy, whatever, just to get to the surface. So, and some of them may not even make it up there. So it could have germinated and you didn't see it. So if you get them about the right depth, then you get a decently healthy plant, usually a really great healthy plant. And I actually scoured the internet to try to find a picture to illustrate this, and there's none for native plants. So I'm going to build the clear little box, and I'm going to make a picture of different different planting depths to see exactly what happens, because you can only find it for ag. I scoured the internet like all earlier this week. I'm like, I need one more picture for this. So I'm going to build a clear little box. I'm going to plant some seeds at different depths, and I'm going to determine it. But a general rule of thumb is you plant them about twice the widest part of the seed, like for the depth. That's a good general rule of thumb. So those little dust seeds, yeah, just go ahead and put them on the surface, because what's half of that? Or twice that. But yeah, and some seeds, if they're just, kind of big, put them on the surface, maybe a light, thin layer, loose soil. But some seeds can get some big, long seeds. You can so take that double length of the longest part. Because if you put them sideways, then the little plant's got to use energy to reorient itself the correct direction anyways. So save them some energy, let them go. Okay. So it's about the right depth as well. And then, you're like, okay, great. So you told me some great tips, but how do I really know for sure, especially if it's like a problem species you haven't been able to get it to actually germinate? So just start thinking about where it lives when it's an adult plant. And that really gives you your best insight on what that needs. And so around here, most of them need winter. Winter is important. And then, what we would consider a regular winter, not these weird warm winters, right? I personally love the short warm winters, but the plants don't. They actually need the cold. It's important for their longevity. So cold stratification is where it's at. So it usually about 30 days in the fridge will mimic that enough. Sometimes it's 60. There's a few that need 6 months, but those tend to be trees. So if you just want, herbaceous forbs, then 30, 60 days, sometimes just 45, check them. You don't want to leave them in too long because then they don't do well. Okay, so the time that they need that cold varies. And then they all, no matter what, need that warm up afterwards. That's what happens. Yeah. Winter and then spring. Everything warms up and we get my favorite time of year. Yes.
Are you familiar with the American basket flower?
Yes.
That's one that I've seen, and I had problems with, and I've heard other people have problems getting them to germinate.
Yeah, I had problems because I got them for one of my 2 seeds from Cherokee Nation, and I'm gonna try again. And I found out later, part of the reason problem may have been they put them all in the freezer for a few weeks before they ship them out to citizens. So I'm gonna try a different source and see if I can figure that one out, because I did it in the fridge for, I think I did 30 days, nothing, and then the seeds just like rotted.
3 or 4 plants come up.
I had 0.
Quite a few seeds, but that was good.
Yeah, and because I really would like to have that, because when I do my vegetable garden, one side is all native plants with flowers that are not edibles, because I live in an HOA neighborhood that everybody wants to spray, and I don't spray, And so the first year, besides bad soil, bad water, there were no bees in the neighborhood. Like, I never saw a bumblebee in the backyard.
So what are you gonna try for the house of flowers?
I think I'm gonna do a whole different seed source, one that I know that hopefully didn't put it in the freezer. And then I want to try to do, I'm gonna take some and do like a 30-day and then maybe a 45 and then a 60-day cold. But I gotta find a good seed source that didn't freeze the seeds first.
Well, the seeds I got, I have a few of my own now that I collected this year, but the first seeds I got were from fellow gardeners.
Yeah,
that
so,
They wouldn't have pulled that like.
right, and so maybe even you could maybe do some cold for a little bit and then follow it up with a heat mat.
Yeah, I wondered if maybe you were talking about scarifying.
Yeah, they might need a little, yeah, it wouldn't hurt to try it.
I wondered if maybe that would help them.
Yeah,
I actually found out one way to treat the snails is using the limestone powder.
yeah,
Yeah, so you don't necessarily have to put it around the plant, but you put it in the pathway that the snail will walk through.
yeah,
So
that would
at the
work.
time when they walk through it, and then they will not make it to the plant.
Yeah, because when I moved from Norman, I brought 2 of those snails and they got squished real quick. 'Cause I saw them, I was like, no.
Eggshells, crushed up eggshells.
That would be strong enough. You need something strong.
Yeah, I mean, I've got a bag of the lime because, I tried to grow squash in the soil. The soil. But yeah, and I devote part of my vegetable garden to just flowering natives to try to get the pollinators to come back, and it's working. We've got native bees living in the ground in the backyard by the shed now, and I'm like, yes. And the bumblebees, that Icomea, that morning glory, I showed you away at the beginning, bumblebees getting fights over those flowers. Yes, come on. And so it's really good because, so I'll plant, random things too, just to try to get more pollinators around, because I was just like, how can everybody spray so much? Because I was like, and when I first got up there, the landlord sprayed the front yard and you couldn't get in the backyard, thankfully, because blueberry bushes. And I'm like, no, and they were fine. But then,
I love it when you see one of those editions and it's called Monarch Landing or something, and there's not a monarch.
right? I know, I'm just like, really? Yeah, but yeah, Alaska, we got my blueberry bushes, so I gotta get a new one or 3. 2 minimum, but cross-pollination, gotta have 2 minimum for blueberries. But so yeah, I try lots of different things, and it just depends on the time. So I'll put together some links for some websites that I have found extraordinarily useful, because before the internet, there's a lot of trial and error, talking to people, buying heavy, thick books. But what's a book, right? I was actually gifted through a chain of people the Baskin and Baskin's Seeds book, and it's a great book, but it's heavy and it doesn't really cover as much as I want because it's been in print for 20-something years. But Prairie Moon Nursery, they're actually based out of Minnesota, but they have a lot of really great seed germination information. It's detailed, it's got lots of pictures, and they've got codes for all their natives that they sell up there. So they're a Minnesota native nursery, but you can still apply a lot of those techniques to our species here. But then when I was doing the Penstemon study for my master's, I came across this Tom Clothier's Garden Walk and Talk, and he has so many more species from all over the US that don't make it into the native plant nurseries. Like, he has done extensive work, and he just makes a blog on the internet, and it's a great website. It's alphabetical by genus then species, and it's great. And if your species isn't in his list, you can start looking at relatives in the same genus, and you'll see patterns, and a lot of them require the same thing. Penstemons do get weird if they're from the Rockies. Outside of the Rockies, there's patterns. Rockies, Penstemons are weird. They need smoke or really cold. So fortunately, there's not a whole lot of native seeds around here that need and require smoke to germinate, because there are some plants that do that, but we don't have them around here. So you just waft some smoke and they're happy. And then if you want really cool graphics, like bar graphs of germination rates, the Sylvan Seeds group, they've got this website, and they focus on temperate deciduous forest species. So there's a lot of trees in their list, but they also have a lot of not trees. And they like talk about if did they scarify the seeds, did they try to add extra light? They have all kinds of different things, and they have just, you go to the website, it's a drop down list, it's alphabetical, it's a huge list. And they do trees or do species even not in North America because it's just temperate deciduous forests and they're looking for patterns. But when you pick a species, they'll even tell, they have a little map, and it'll have a little yellow point telling you where they got their seeds from, and there's some on there from around here. So, but they have a global list. So you know, you got to think about that right place. So if you're going to go the easy way and just stick them outside, be patient, right? Think about what they need. Make sure you go water them enough, but not too much. Because I did try to put my pollinator garden in my yard in Norman, and then it turned into an ice rink 2 inches thick. So failure, but that's okay. And also think about when the big trees start throwing their shade. Is your area going to have enough sunlight? Because a lot of our native, or at least ones I like, need full sun. So that means one side of my yard is off limits because there's a big silver maple I'm not allowed to cut down. I want to, but I'm not. So the other side, it's fair game for those big full sun species. But then if it needs shade, hey, I've got over half the yard ready. And then the soil, you think about your soil, think about the soil that those plants would normally grow in out there in the wild, not in our yards. All right. If you want to do them inside, here's the sassafras seeds before they went to the cold frame. Nice, 1 seed per cell. Life of a grad student, I don't miss that part. But like I said, I really like vermiculite just because of the sterility. You can order big bags of it online, really cheap. You can buy it at a lot of the nicer nurseries. They'll have just straight vermiculite. You can even mix it in with your potting soil to help loosen it up, make it airy, light. That way the seeds have less, or the little plants have less to push against when they come up. And you can mix in sand, but again, I don't recommend straight sand just because of the lack of water retention. That's the properties of sand. And then if you want to throw them out in a cold frame, that's great. It's easy, it's fun. Cold frames are nice and useful, except when Alaska visits, because nothing survives the cold frame. Because my tuberoses, I'm gonna order new bulbs. I put those out in that cold frame I told you about. When you do a cold frame, make sure it's anchored, because you know the wind blows here, and this happens.
Oh, no.
Yeah, this happens. So, and I even had it staked with the stakes it came with from the inside, and I had bricks on the 4 corners on the top, and the wind still got it. So make sure you're anchored in really well if you are using a cold frame, especially a lightweight aluminum style like that. Now the cold frame, just if I put it against the south side of the house, it's usually okay from wind, but not sub-zero temperatures.
Are you sure there was not a squirrel there?
It could have been a goose. This was out at those large greenhouses on the south end of the OU campus. It was just open, flat space where I decided to put it because I wanted natural winter condition, temperatures especially. So yeah, that happened. I about cried. That was like 200 seeds too. I found most of them. So,
Wow.
I was a dedicated grad student before the snails. I mean, that project was doomed from the start. Wind and then snails. But yeah, but now I've upgraded. I have a dedicated fridge in my lab. It doesn't even have a freezer. It is a straight fridge, but I got it for free because somebody was throwing it away out of the student union. I just had to fix the power cord. So.
Instead of a beer fridge or next to the beer fridge.
Right? Yeah, I've got the garage fridge too for backup freezer space and extra beverages. But yeah, so I've upgraded. And so I can, like I said, I can fit full flats in there. So that's what I've got going on in there actually right now. So yeah. And then oh yeah, there's that bit about sand. I already talked about that. So yeah, that's all I got. I think I went a little long, sorry. But thank you for listening. I am on iNaturalist. I help out with BioBlitzes all the time because that's my friends down there in Norman doing those. So my username on iNaturalist is just J Messick. And if you want to email me, my email is jmessick1, the number 1, @uco.edu. I've also got cards if it helps for that. Yeah, if you leave off the 1, somebody else gets it. It's not me. I know who it is. It's a family member, but it's not me. Because younger ones have gone to UCO. For some reason, they got to go to UCO, didn't have to go to OU. But thank you for listening. And
Questions for Dr. Messick? So Jenna,
yes,
thank you very much.
yeah, no problem.
As I was talking earlier about this Douglas Talamy,
Mhm.
he's really a voice that all of us that are in the native plant interest group should probably be aware of.
Yeah.
His first book was Bringing Nature Home. Anybody in the audience that's not familiar with it, I strongly recommend it. It's an easy read. It's not scientific, but it absolutely makes sense. So on behalf of the Central Chapter of the Oklahoma Native Plant Society, I hope you'll read it. It's a very enjoyable
Well,
read.
thank you very
And we're
much.
also going to give you a complimentary membership for the next year to ONPS. I hope we'll see you around.
Yeah, that'll be fun. Thank you very much.